When you see neurons portrayed in Hollywood movies, this is what you usually see, sparks of light flying between cells. Not quite what's going on. What we're going to talk about today is how nerve cells conduct what's known as an action potential, what that action potential is, and how it conveys information from one cell to the next. Here you're seeing a pretty typical motor neuron. You've got a cell body that contains the nucleus, and then attached to that, going off to the right in this case, you've got a very long projection known as the axon. This is not to scale. The axon can be remarkably long. It can be over a meter in length in some cases. Note that we also have these structures shown in green that are called dendrites that carry in action potentials or electrical signals towards the cell body. Information in the terms of electrical signals always travels in the same direction. It travels down dendrites towards the cell body. It travels away from the cell body down the axon. This one cell can be attached to hundreds or thousands of other cells. So we have these action potentials or electrical signals coming down through the dendrites, and again, they may be collected from several different cells. They head towards the cell body. Now these electrical inputs are going to change the electrical properties of the cell body. If there's a voltage difference between the inside of the cell and the outside of the cell above a threshold level, then this cell will fire off its own action potential. This is determined here. So if we get up to a threshold voltage in that area, an area known as the hillock, then this cell will fire off one discrete electrical signal. And that electrical signal or action potential will travel down the axon to the end of the axon. And you can see here that once again, this cell can connect to lots of different cells. We've got several terminals there that can connect to different cells. Neurons, and especially axons, are quite often compared to copper wires, the kind of wires that we use to conduct electricity in our homes. This really isn't quite an accurate comparison. In a copper wire, we've got atoms of copper that are staying put, they're not going anywhere, and electrons are bouncing from one copper atom to the next. So somewhere there's a generator that is spinning magnets, and the magnets are pushing electrons along this chain of copper atoms. In neurons, though, what's happening is ions, so remember those are charged atoms, are being moved back and forth across the membrane of the cell to set up a charge differential across the membrane. That charge differential is a voltage. And then that charge differential is going to move down the length of the axon or the dendrite. So it's this movement in potential that occurs. This is done by moving ions, and the ions can be moved through a number of different channels. Leak channels, ligand-gated channels, mechanically gated channels, voltage-gated channels, and also the sodium chloride pump is used to set up ion concentration gradients across the membrane. And we'll come back to each of these and look at them in detail. Here you're seeing a representation of the plasma membrane of a neuron. So we have our phospholipid bilayer in the middle of the diagram. On the left, we have the exterior of the cell, and on the right, we have the interior. Note that within the interior of the cell, we have some very large proteins that are represented by the letter A here. These proteins have multiple negative charges. Now, because these proteins are large, they can't readily cross the plasma membrane, so they're going to stay put inside the cell, and they're going to give the interior of the cell this base negative charge. But we don't just have these negatively charged proteins, we also have a lot of potassium. Remember that potassium is represented as K on the periodic table. Potassium ions are positively charged. The potassium ions can leave the cell through channels, and you can see a channel at the top of the diagram there that's known as a potassium leak channel. This is a passive process, and it's an example of facilitated transport. So potassium is built up in high concentrations inside the cell, it can diffuse down its concentration gradient, but only if those channels are present and they're open. Remember that when we're talking about ions moving across a membrane, we have to consider two things. We have to consider the concentration gradient, and we also have to consider the charge gradient. So ions that have the same charge are going to repel each other. Potassium is not going to leave the cell if the exterior of the cell is very positively charged, because that positive charge will repel the movement of the phosphates. On the exterior of the cell, you can see that we have a lot of sodium. And notice that a lot of the sodium is stuck to the outer leaflet of the membrane. So those phospholipids that make up the membrane have a hydrophilic head, and that has both a negative and positive charge to it. The negative charge of the phosphate within the hydrophilic head will stick to those sodium ions. If there aren't any sodium channels, sodium can't get into the cell. It's just going to stick to the outer part of the membrane. Now there's also pumps that we'll talk about in a moment that allow the movement of potassium and sodium, in addition to these passive channels. Let's take a look at those pumps and channels. Leak channels are exactly what they sound like. They allow ions to leak out of a cell or into a cell. This is an example of facilitated transport. It doesn't require any energy, it doesn't require ATP, and the movement can occur quite quickly. But the channels are specific to specific ions. We have potassium leak channels that will allow potassium out of the cell, and we have sodium leak channels that will allow sodium to move into the cell. Note that these channels can be opened and closed. It's also important to note that the plasma membrane of a neuron has far more potassium channels than it does sodium channels. So the membrane, the plasma membrane, is more permeable to potassium than it is to sodium. Channels that can be opened or closed are referred to as gated channels. A ligand is a molecule that can bind to an enzyme or a channel and either activate it or deactivate it. In the case of a channel, the binding of this molecule is going to cause the channel to open if it's closed, or perhaps it'll cause it to close if it's already open. Neurotransmitters and hormones are good examples of ligands, and the channels that are controlled by these ligands are known as ligand-gated channels. Some channels can be activated or deactivated by a number of rather different stimuli. What you're seeing here is an example of a channel that's found in pain receptors within your skin. If the skin is exposed to a strong acid, what happens is the high concentration of hydrogen protons causes a change in the shape of the channel. It's usually closed, but in a high acidic environment it opens up, and it allows positively charged sodium and calcium to rush into the cell, change the membrane potential of the cell, and cause the cell to send off an action potential. It responds the same way if it's exposed to capsaicin, which is a molecule that's found in hot peppers. It's what causes the pepper to burn. What's happening here is the capsaicin is going to bind as a ligand to this channel, cause the channel to open up, and again cause sodium and calcium, which is piled up outside the cell, to flow in, cause the cell to send off an action potential. If the skin is heated to high temperatures, if you touch something hot, then what happens is this channel is going to change shape. It's going to change shape, it's going to open, it's going to allow sodium and calcium in, and it's going to depolarize the cell and cause it to send off a signal. One more thing can set this off. If there's an increase in osmotic pressure around these cells, that will also trigger this cell in exactly the same way. So if you have an inflammation of an area, if you have a pimple, as you can see here, that puts pressure on the cell because we have lots of interstitial fluid around it, Again, that changes the shape of the channel. So all of these different stimuli will cause the channel to open, and it'll trigger that pain receptor, which will then send a signal back to the brain, and your brain will interpret that as pain. Mechanically gated channels respond to forces, usually stretch, within the membrane that they're embedded in. Think of the plasma membrane as kind of a trampoline. If you stand on a trampoline, of course, you stretch that membrane. Same thing happens to the cells in your skin, for instance. If someone applies a strong touch, a strong force to your skin, then that is going to stretch out the plasma membrane of cells within the skin, including touch receptors. So let's say again that we're dealing with a touch receptor in the skin. If we have stretching of the plasma membrane, it's going to pull in all directions on that channel. And when that happens with a mechanically gated channel, the top, this kind of lid-like structure, opens up. So it causes this channel to become leaky, and then the calcium and sodium ions that are piled up outside the cell will flow into the cell. And this causes the interior of the cell to become more positive. We call that depolarization. I'll come back to that in a bit, but that's going to cause this cell to send off a trigger. This is how touch receptors work. It's also how the receptors in the ear work. We can also have channels that are voltage gated. Voltage refers to the charge differential across the membrane. It works the same way for batteries. If you have a battery that can generate a lot of volts, what that means is there's a big difference in charge between the positive terminal and the negative terminal. So on the left, we have a neuron that's at rest. And neurons that are at rest usually have a membrane voltage of negative 70 millivolts. We're comparing the charge within the cell to the charge outside the cell. The negative tells us that this inside of the cell is negative with respect to the outside. Now notice on the left we've had a change in that membrane potential, and now the voltage is negative 50 millivolts. We have less difference between the inside and the outside. The inside is still negative, but the difference isn't as great. That change in membrane potential causes this channel to change shape. This is a voltage-gated potassium channel. It was closed at minus 70 millivolts, but now at minus 50 millivolts it opens up, and that allows potassium to rapidly escape from the cell. Finally, and very importantly, we have the sodium-potassium pump. So as we'll see, when an action potential passes along a neuron, what's happening is voltage-gated and other gated channels are opening up and closing, and that's allowing certain ions to move in or out of the cell. But after that action potential passes by the membrane, we have to re-establish the resting potential, the potential that we have in the membrane of a neuron that's not actively firing. That is accomplished by the sodium-potassium pump. This is a protein that's embedded within the membrane. It can grab on to potassium and also to sodium, and it can change shape, and by doing that, move the ions across the membrane. So what it does is it grabs onto sodium ions that are found within the interior of the cell. They have little slots that they stick into, and the molecule can grab onto 3 of them. ATP binds, it donates a phosphate, and that causes a change in the shape of this protein so that now the sodium can be moved to the other side and dumped into the exterior surrounding the cell. Next, what happens is 2 potassium ions come along and they bind to slots in the molecule. The removal of the phosphate causes a change in shape again. It goes back to how it started, and it dumps those phosphates into the interior of the cell. Now, the really important thing to note here is that for every molecule of ATP that's used, 3 sodium ions are moved out of the cell, but only 2 potassium ions are moved in. So 3 positive charges are moved out, 2 positive charges are moved in. And that means that the interior of the cell is negative with respect to the outside. Now this can be a bit confusing. We're not necessarily suggesting that the inside of the cell is negatively charged, but it's negative relative to the outside. It's less positively charged inside than it is outside. This is an active process. This is active transport. This requires a lot of ATP, and this is why nerve cells are very, very hungry for energy. To sum up then, if we look at a neuron that's at rest, it's not actively conducting an action potential, we have the sodium-potassium pump hard at work all the time, moving sodium out of the cell, moving potassium in. We have leak channels that can allow both sodium and potassium to move across the membrane, but there's a lot more potassium leak channels than there are sodium channels. What this is going to do is it's going to establish this negative 70 millivolt resting potential. The inside of the cell is more negative than the outside. Now keep in mind in this diagram, we're not seeing those negative proteins that we talked about earlier. This is what we have for a resting membrane. Hopefully from chemistry you remember that if you see square brackets around something, that's shorthand for concentration. So we're looking at the concentrations of 3 different ions that are found on the outside and inside of a dendrite. So compare potassium, for instance. We have 140 millimolar concentration of potassium on the inside of the dendrite and only 5 millimolar on the outside. So a lot more potassium inside than outside. It's the reverse for sodium. We have more sodium on the outside than inside. We also have more chloride on the outside than we do on the inside. Now each one of these ions has a charge value of 1. It's either going to contribute 1 plus charge or 1 negative charge. If you've gone ahead and done the math, first of all, bravo, and secondly, you probably noticed that it doesn't add up. Remember that we also have negatively charged proteins within the cell, and those proteins have multiple negative charges, and it's variable from protein to protein. Let's look at a couple more hypothetical examples before we move on to investigating what happens in living nerve cells. Let's say we have a container and we divide the container into 2 chambers using a barrier of some sort. Embedded within the barrier, we have channels that will allow potassium ions to move, but nothing else. Now let's say that we take a solution that's 140 millimolar of potassium chloride, we add that to our inner chamber, and to our outer chamber we add a solution that has a concentration of 5 millimolar potassium chloride. Potassium chloride is a salt. When you add it to water, it's going to dissociate into potassium ions, which are positive, and chloride ions, which are negative. Now what's going to happen is the potassium is going to move through those channels down its concentration gradient. So it's going to move from the inner chamber to the outer chamber. It will continue to do that until the outer chamber becomes positive enough that it repels the motion of additional potassium. So remember, the chloride is staying behind, and that means the inner chamber is going to become more and more negative while the outer chamber becomes more and more positive. However, as mentioned, we will get to a point where the potassium stops moving. It's going to be repelled by the potassium that's in the outer chamber, and the potassium that's inside the cell, or in the inner chamber, is going to be held in place by its attractions to the chloride. We reach an equilibrium where we might have some movement back and forth, but the charge, the voltage, the differential between the 2 chambers doesn't change. And we could stick in a little electrode and we can measure that. And for this example, we have an EK, so an equilibrium of potassium, of minus 90 millivolts. Now let's do something similar with sodium chloride. Sodium chloride is also a salt. If you add it to water, it's going to dissociate into sodium, which is positive, and again, chloride, which is negative. So we add 150 millimolar of sodium chloride to the outer chamber. And we're going to add 15 millimolar of sodium chloride to the inner chamber. Once again, we have a barrier between the two chambers, and we have channels that will allow sodium to move, but they won't allow the chloride to move. The sodium is going to move from the outer chamber into the inner chamber, and that's going to make the inner chamber more and more positive because the chlorides are going to be left behind in the outer chamber. When we reach equilibrium, we have a value in this hypothetical example of plus 62 millivolts. So our ENA, our equilibrium for sodium, is plus 62 millivolts. Remember that the plus or minus sign always refers to the inner chamber. We're looking at the inner chamber or inside of the cell relative to the outside. So in this case, we've got a plus sign because the inside of the cell is positive compared to the outside of the cell. Now let's combine these two gradients. Imagine we have a hypothetical cell that has a lot of sodium on the outside and a lot of potassium on the inside. If we open up channels that allow sodium to move, the sodium will move from the outside to the inside, and the membrane potential will move towards the equilibrium value for sodium. Likewise, if we close those sodium channels and we open up potassium channels, then potassium will move out, and the membrane potential will move towards the equilibrium value for the potassium. Again, this can happen very quickly, and it happens due to the opening and closing of gated channels. Membrane potentials can be measured. We can estimate them if we know the concentration of sodium and potassium and other ions inside the cell or out, but we can actually physically measure them. There are microelectrodes that are very, very thin. They're wires that are thinner than a hair, and they can be injected into tissues or even into cells, and then we can compare the charge within the cell to the charge of the interstitial fluid outside the cell, and we can get a real-time reading of the membrane potential. If gated channels within the membrane of a sensory cell or neuron open up, they allow ions to move, and that's going to cause a change in the membrane potential. A small change in the membrane potential is referred to as a graded potential. If it reaches a certain threshold, it can cause the cell to send off its own action potential, as we'll see. In a hyperpolarization, the interior becomes more negative. So we could have negative ions moving into the cell, or we could have positive ions moving out of the cell. So the interior becomes more negative relative to the exterior. In a depolarization, we have the interior becoming less negative or more positive. So what's happening here is we have positive ions coming in, sodium for instance, or we could have negative ions leaving the cell. Here's an example of a hyperpolarization. We're starting with a cell that has a resting potential of about minus 70 millivolts. Note that there's a threshold value. This value here is the value that we'd need to have for the membrane potential in order for an action potential to be fired off. Essentially, if our resting potential were to become less negative to the point where it reached this threshold, then voltage-gated channels would open up within the membrane. But that's not what's happening. Instead, the membrane potential is moving in the other direction. It's becoming more negative instead of less negative. This can be caused by the outflow of cations, which are positively charged, or the inflow of anions. A hyperpolarization moves the cell further away from that threshold value. In a depolarization, we have the inflow of cations or the outflow of anions, or both. And what's going to happen is the interior of the cell is going to become less negative compared to the exterior. If we get up to our threshold value, then we can have an action potential being produced. So what causes these initial changes in membrane potentials? Well, we talked about a few examples already. Remember mechanically gated channels. Mechanically gated channels open up if pressure is applied to a cell. So basically what happens is the walls of the channel are stretched and the little lid on the top no longer covers the interior of the channel and stuff leaks in. These channels will specifically allow sodium or calcium to enter the cell. And sodium and calcium, of course, are positively charged. So these positively charged ions rush in and they cause a depolarization. They make the interior of the cell more positive with respect to the exterior. If we pass our threshold, the cell will then fire off an action potential. The opposite can occur. We can have a cell that's inhibited by the movement of ions. So imagine that we have a ligand-gated channel that binds to perhaps a hormone or maybe a neurotransmitter. And what this ligand does is it causes the channel to open, but in this case it's going to allow chloride to move in. What that's going to do is it's going to hyperpolarize the cell. It's going to make the interior more negative relative to the exterior. And now the cell becomes less likely to send off an action potential. We're basically inhibiting the cell. We're removing it further from that threshold voltage. Imagine a neuron that synapses with several other neurons. It's getting signals through dendrites, and those signals are passing on to the body of the cell. Now, the signals that are received cause a depolarization in the cell. They cause sodium ions, for instance, to rush into the cell and make the interior less negative. So our cell is going to start out with a resting potential of minus 70 millivolts. The thing is, though, unless that depolarization reduces the negativity of the interior of the cell up to our threshold, which is about minus 55 millivolts, nothing is going to happen. The cell is not going to be stimulated by a sub-threshold stimulus to fire off its own action potential. So you can see in this example here, we've got 3 stimuli that are coming in, and each one is a bit larger, but even stimulus 3 is not large enough to bring us up to our threshold. In that last example, we had discrete stimuli, and there was enough time between the stimuli for the sodium-potassium pumps and leak channels to bring the cell back to its resting state. Now imagine if the stimuli occurred immediately, one after the other, and there wasn't enough time for the cell to return to the resting state. What can happen is we have a process called summation. In this case, we have stimulus 1, and that's going to cause a depolarization, and sodium ions are going to rush into the cell and make it less negative. Before the leak channels and sodium-potassium pumps have an opportunity to correct that, a second stimulus hits and more sodium rushes into the cell, and the cell becomes even more positive. You can imagine that if you have several stimuli that occur one after the other, this could potentially bring the cell up to threshold levels. So the effects of the stimuli are being summed, they're being added together. Remember that hyperpolarizations bring the cell further away from the threshold, whereas depolarizations bring the cell closer to the threshold. And if those depolarizations are very strong, or if we have a lot of small ones that are additive, and we make it to threshold, then voltage-gated channels are going to open up, and that's going to allow the rapid influx of sodium. This is what that looks like in graph form. So you can see we have time on the bottom in milliseconds, and then we have membrane potential along the y-axis. We have our resting potential of minus 70 millivolts. We have graded potentials that bring us up to threshold, and then all of a sudden we have this rapid change in the membrane potential, and the membrane potential actually becomes positive. And then it drops back down, and it takes a little while to be corrected back to our resting potential. An action potential is all or nothing. Once a cell reaches the threshold, it's going to fire off this very rapid depolarization, followed by a rapid hyperpolarization until it comes back to its resting potential. So let's take a look at what's going on here. First of all, we start out with a cell that's at its resting potential. This is where nerve cells are going to be if they're not actively conducting signals. So the resting potential is minus 70 millivolts, and this is maintained by the sodium-potassium pump. Now we're going to have inputs, maybe from mechanical gated channels, for instance, or maybe inputs from other cells that synapse with this cell. And they're going to cause small depolarizations, and that may bring the cell up to threshold. Once it gets up to threshold, very suddenly, voltage-gated sodium channels will open. They'll pop open and they'll allow a whole lot of sodium to rush in. Remember that sodium is in high concentrations outside of the cell. That causes the membrane potential to become positive. What's going to happen next is repolarization, and this is due to voltage-gated potassium channels opening. They're going to open, and they're going to allow potassium out of the cell. Potassium, as it leaves the cell, carries with it positive charge, and that causes the membrane potential of the cell to drop and become negative again. And it actually undershoots the resting state value. The resting potential is going to be reestablished once again by sodium-potassium pumps. The action potential always has the same profile, and it's always all or nothing. So if we get up to threshold, and threshold is minus 55, then we're going to have this rapid depolarization that brings us up to plus 30. And then we're going to have a repolarization. So if we have a sub-threshold stimulus, no action potential. If we have a very short-lived but supra-threshold stimulus, we're going to get an action potential. If we have a sustained stimulus, so you can see in the last example here, we've got a stimulus that's very strong and it's sustained for a fairly long period of time, that's going to result in multiple action potentials if that stimulus keeps occurring. However, we're going to get distinct action potentials that all have the same shape. They have the same profile to them. Let's take a look at this again, this time just focusing on the voltage-gated channels. So we start at number 1, that's our resting state. And remember that that resting state is maintained by the sodium-potassium pumps. They're moving sodium out of the cell, they're moving potassium into the cell, they're setting up this gradient that can be used to do work. And in this case, that work is creating an action potential. We have depolarization occurring, and that can be due to some sort of outside stimulus, but it's going to cause some sodium channels to open and allow sodium into the cell, and we get a depolarization. If we get up to threshold, then we have all of these sodium voltage-gated channels opening up, and that causes the rapid influx of sodium. And that's part 3. That's the rising phase of the action potential. We get up to that plus 30 level, and then what's going to happen is the sodium channels, the gated sodium channels are going to close, and now the voltage-gated potassium channels are going to open. They're going to allow potassium to rush out of the cell. That's going to cause this falling phase of the action potential. Now, part 5 there, we get what's called the undershoot. We actually overshoot that -70. And the reason for that is these voltage-gated potassium channels react fairly slowly when it comes to closing. They don't close immediately, and they allow out a little more potassium than you would expect them to. So we get this undershot before those potassium voltage-gated channels close completely. But then, of course, our sodium-potassium pump is going to bring us back to our resting potential, and it's going to establish those electrical and concentration gradients across the membrane. But what causes an action potential to move, and why does it only move in one direction? Remember that the action potential in an axon moves away from the cell body. It's always propagated this way. Well, let's take a look at these 3 diagrams here. In these diagrams, plasma membrane that's at its resting state is shown in red. Plasma membrane that is depolarizing, so undergoing the rising phase of an action potential, is shown in blue. And membrane that is repolarizing, so it's in the falling phase, is shown in green. In the top diagram, at the left-hand side of the axon, we have depolarization occurring. So the membrane was depolarized above threshold, and now we have voltage-gated sodium channels that have opened up, and they're allowing the rapid influx of sodium. Now that sodium is going to diffuse through the cytosol in both directions, and some of it is going to diffuse over here. And when it diffuses over here, it's going to depolarize that section of the membrane beyond threshold, and it's going to cause sodium channels, the voltage-gated channels in that region, to open, and sodium is going to rush in, and we're going to get a new depolarization. That's what you're seeing in the second diagram. So this new polarization is going to cause sodium to rush in, and it's going to rush in both directions again. It's going to travel through the cytosol, and to the right, it's going to cause a new depolarization, and that's what you're seeing in the bottom diagram. However, sodium that diffuses towards the left is going to have no effect. And that's because that section of the axon is undergoing repolarization. So we got up to our plus 30 millivolts, and that caused the sodium voltage-gated channels to close and the potassium voltage-gated channels to open. So potassium is rushing out, and even though sodium is entering into that area, it's diffusing into that area, it can't open the sodium channels, and sodium can't rush in. We can't get depolarization. So that section that's shown in green is in a refractory period. It can't be stimulated, and it can't be stimulated until it returns to the resting potential. So what that means is that even though sodium is diffusing in both directions, we can only have the propagation of the action potential in this direction. Electrical signals, depolarizations, are going to travel along the dendrites to the cell body. If these depolarizations bring the axon hillock area up to threshold, then it's going to fire off an action potential. And the action potentials are going to be discrete signals that travel in one direction away from the cell body. A neuron will have an absolute refractory period and a relative refractory period. During the absolute refractory period, there is no way that the cell can fire off another action potential. So this is the rising phase of the action potential and the majority of the falling phase. It's just simply not possible to stimulate the cell to set off another action potential. After that, we have what's called the relative refractory period. During the relative refractory period, it is possible for the cell to send off a new action potential, but it's very unlikely, especially during that hyperpolarizing phase, that undershoot. During that undershoot, the cell is further away than normal from the threshold. So we'd have to have very strong signals, very strong depolarizations to bring us back up to threshold. Axons that are especially long are often myelinated. They're covered in a material known as myelin, and this consists of multiple layers of phospholipid, and it acts like an insulator. Now, it doesn't run continuously along the axon. There's nodes, known as nodes of Ranvier, where the axon is naked. It doesn't have this myelin. Those are the only regions where the exchange of ions can occur between the interior of the axon and the exterior of the cell. This is where an action potential can be generated by the means that we've already discussed. So we have action potentials that are generated within the cell body. They travel down the naked portion of the axon, and when they get to one of these myelinated areas, the sodium that rushes in is going to push sodium in front of it through the axon. So that sodium is going to be forced by repulsive forces and by diffusion to the next node. And at the next node, we have a new action potential being generated, traveling down the naked portion of the axon until it gets to the next myelinated area, where once again, we're going to have sodium being forced through the cytosol of the axon to the next node. This is a much faster mode of transmission. So in axons that have to conduct signals, meters or more in distance, we have this saltatory conduction. Saltare in Latin means to jump. So basically this electrical signal is jumping from node to node through the axon. We've talked about how signals are transmitted within a neuron, but how do neurons connect to other neurons and other cells? Well, they do this at a synapse. So a synapse is a junction between one neuron and another neuron, or between a neuron and an effector of some sort. There are two types of synapses. We can have a physical space, and then we have a signaling molecule, a neurotransmitter that will cross that space. Or we can have direct connections and gap junctions that allow ions to move from one cell to the next. In an electrical synapse, the cells are physically attached to each other, and gap junctions form a direct connection between the cells. Ions can pass from one cell to the next directly. So if one cell becomes depolarized by the influx of sodium, the sodium will move through gap channels into the next cell and depolarize it as well. These gap junctions allow for ions and other signals to move in both directions. So this is a junction that will allow information to potentially move in both directions. We can also have chemical synapses. In this case, we have an actual space between the two cells that are interacting. We have a neurotransmitter that's released at the end of the axon, and that neurotransmitter is going to diffuse across that space, the synaptic cleft, and then it's going to bind to a receptor on the other side of that cleft. So in this case, we have neurotransmitter always being released from one side, and we have receptors only found on the other side of the gap. So in this case, the information is only transferred in one direction. Chemical synapses are more easily regulated by the body, and they're also more common than electrical synapses. So we'll talk about how they work in a bit of detail. At the end of the axon, we have structures known as synaptic end bulbs, and we can have several of them. The synaptic end bulbs contain vesicles that contain neurotransmitter. When the action potential gets to the end bulb, it's going to activate voltage-gated calcium channels. These channels will open and they allow calcium to enter into the end bulb. When calcium comes into contact with these vesicles, it makes the vesicles able to bind to the plasma membrane. They can't bind to the plasma membrane for reasons we'll talk about in a second unless calcium is present. So now that the vesicles are able to fuse with the membrane, they'll do that and they'll dump out the neurotransmitter through exocytosis. The neurotransmitter diffuses across that tiny little gap and it will bind to neuroreceptors on the other side of the gap, so on the postsynaptic side. Typically, the receptors are channels. The channels will open. They'll allow sodium, for instance, to flow into the next cell and bring about an action potential in the next cell. And here we can see that again. So again, an action potential is going to come down the axon to this end bulb. That's going to cause calcium to rush into the end bulb. That's going to cause these vesicles to bind with the membrane and dump neurotransmitter into the synaptic cleft. Neurotransmitter diffuses across the cleft. It's going to bind to these ligand-gated channels, which are usually channels for sodium, and then the sodium will rush in, that'll depolarize the next cell, and of course if it brings it up to threshold, it's going to fire off its own action potential. Calcium plays a number of important roles within the body, and one of them is allowing membranes to bind to each other. The membranes bind to each other using a molecule known as cadherin. So what you're seeing here, we have two membranes, and we have these cadherin molecules sticking out from each of the membranes. The cadherins can stick to each other. This is the first step in pulling two membranes close enough so that they can fuse. Note that the cadherins on the left are rather flaccid. They're quite floppy. When calcium binds to them, they stiffen up and they form this rod that can connect to other cadherin rods. So again, if calcium isn't present, membranes can't bind. In the synaptic end bulb, if calcium isn't present, the vesicle can't fuse to the plasma membrane and it can't dump the neurotransmitter into the synaptic cleft. A neurotransmitter can have either an excitatory or inhibitory effect. So we've already looked at the excitatory, and we'll just talk about it again. Imagine a situation where a neurotransmitter is released into the synaptic cleft, it crosses to the other side, and it binds to ligand-gated sodium channels on the postsynaptic cell. Causes those to open, sodium rushes in, depolarizes that cell, and causes it to produce its own action potential. That would be an example of an excitatory response. However, neurotransmitters can also be inhibitory. Let's imagine this time that a neurotransmitter crosses the synaptic cleft and binds to a ligand-gated chloride channel on the postsynaptic cell. That allows chloride ions into the cell. That's going to hyperpolarize that cell. Or let's say that our neurotransmitter binds to ligand-gated potassium channels. They open up and they allow potassium out of the cell. The cell is going to leak positive charge. Again, that's going to cause the cell to hyperpolarize. If the cell hyperpolarizes, it moves away from that threshold. So it's less likely to send off an action potential. Any particular neuron can connect to hundreds of other neurons. Some of them are sending excitatory signals, Some of them are sending inhibitory signals. Whether or not that particular cell is going to fire off its own action potential results from the sum of inputs that are inhibitory and excitatory. One of three things can happen in the postsynaptic cell. First of all, nothing might happen. If nothing happens, it's because the cell did not depolarize sufficiently. It didn't get to that threshold of negative 55 millivolts. It could be that the signal that was received was far too weak, or it could even be that the inhibitory and excitatory signals from several different neurons canceled each other out. The second possible response is that we get the production of an action potential. And that, of course, is only going to occur if we have sufficient depolarization to bring the cell up to threshold. Finally, we could have inhibition occurring. So it's possible if we have a strong inhibitory reaction in the cell that the potential could drop well below the resting potential. The further it drops from resting potential, the harder it's going to be to excite that cell and bring it back up to threshold. Whether or not a postsynaptic cell is going to depolarize sufficiently to produce its own action potential depends on the concentration of inhibitory and excitatory neurotransmitters within the synaptic cleft. If we have a lot of excitatory neurotransmitters, we're going to have more of the sodium channels opening up, and we're more likely to reach threshold. If we have lots of inhibitory neurotransmitter floating around in the synaptic cleft, then we're going to have channels open up that will bring about hyperpolarization. So the end result is a summation of the effects of both of these types of neurotransmitters. This summation can be spatial or temporal, and we'll talk about what that means now. It's important to note that neurotransmitters don't persist for very long in the synaptic cleft. They are very rapidly broken down by enzymes and then transported back to the end bulb where they can be repackaged and reconstituted into vesicles. The reason that the neurotransmitters have to be removed is because the signal would never disappear if they weren't. So let's say we had a lot of excitatory neurotransmitter within a synaptic cleft. If that neurotransmitter just sat there, it would continue to excite the downstream neuron. It would never turn off. Let's imagine a case where we have one neuron, neuron C, in our diagram, and then we have two neurons that connect to it, A and B. And let's say that both of these are excitatory. They're dumping excitatory neurotransmitter into the synapse. You can see down the bottom here that we have A sending off a signal, neurotransmitter is released, and that causes a slight depolarization in C, but it's nowhere near threshold level. Now also, in that short little time period between the excitation of A and the excitation of B, a lot of that neurotransmitter has been removed from the synapse. So when B fires off its neurotransmitters, it has the same effect. We get a slight depolarization, but not enough to bring us to threshold. Now imagine that we had a lot of neurons connected to C. So maybe we've got 24 now. And they're all firing off lots of neurotransmitter, and before that neurotransmitter can be removed, new neurotransmitter is coming in, and that is piling up within the synapse. It's going to stimulate a lot of gated sodium channels to open, and we get to our threshold level. That's spatial summation. So spatial summation, we're talking about the release of neurotransmitter from several different end bulbs. In temporal summation, reaching threshold is the result of very rapid releases of neurotransmitter by one neuron. So you can see at the top there we have our presynaptic neuron, we have our postsynaptic neuron, And this presynaptic neuron is firing off an action potential one after the other, and it's constantly releasing neurotransmitter into the synapse. It's releasing it so quickly because there's so many of these action potentials that the level of neurotransmitter never has a chance to drop, and we have lots of sodium channels open, bringing the postsynaptic neuron up to threshold. Incidentally, this is called temporal summation because temporal means time. This has to do with the fact that the inputs are coming one after the other very quickly. Because there are so many ways that this system can be regulated, this leads to a lot of complexity and subtlety, and that allows for a lot of complex interactions between nerves. Coupled with the fact that the circuitry, the connections, can be complex themselves, this brings about the possibility to do very complex things like thinking. So the neurotransmitters can be excitatory or inhibitory, or in some cases they can be both. So some neurotransmitters can be excitatory in some parts of the central nervous system and inhibitory in other parts. It just depends what receptors are present on the postsynaptic cell and what those receptors do. There are a number of neurotransmitters, some of which we'll talk about in more detail, but acetylcholine is a big one. That's the most common one. Norepinephrine and epinephrine we talked about briefly when we talked about the endocrine system, and we'll talk about those again when we talk about the autonomic nervous system. Dopamine and serotonin, you might have heard of those before. There are a number of antidepressant drugs, for instance, that help regulate the level of these two neurotransmitters and make sure that they're at appropriate concentrations within the synapse. In summation, see what I did there? If the excitatory effect is greater than the inhibitory effect, then we can have the depolarization of the cell occurring. If we get up above threshold, so we have a super threshold excitatory postsynaptic potential, then that nerve cell is going to fire off an action potential. It's going to fire off a signal to the next cell in the sequence. If we have an inhibitory effect that's greater than the excitatory effect, the opposite occurs. The membrane hyperpolarizes. We get this inhibitory postsynaptic potential. And that means that the cell is now less likely to send off an action potential, even if it receives inputs from other neurons. Your brain is a lot like a computer. So in a computer, there are millions of transistors, and transistors are basically just switches. They're on or they're off. They either send a signal or they don't send a signal. If you're familiar with binary code from computing, that's what the ones and zeros are. Ones are on, zeros are off, and this is instructions for turning on and off transistors. In your brain, of course, you have neurons, but they work the same kind of way. They're either on or they're off. They're sending action potentials or they're not. They're being actively engaged or they're being inhibited. Now, where the magic comes is how those on-off switches or those transistors or neurons are arranged. And if you have millions of transistors or millions of neurons, you can do some pretty amazing things. So it's not something we'll talk about in a lot of detail. It's a little beyond the scope of this course, but let's just look at a few patterns that neurons can be arranged in. We can have what's called a diverging circuit, where we start with one input, and this is going to be magnified because that one input will activate two cells, each of those will activate two cells, etc. So we'll have a small input with a large output. Or we can have the opposite. We could have several neurons connecting to one neuron. So now we're collecting stimulus from a large area, and we're pooling that into one output. We can have what's called a reverberating circuit. So that's where we have one input, and that's going to activate a series of cells, and the cells at the end of that series go back and activate the first cell. So this loop will keep firing. And then we have what's called parallel after discharge circuits, where our initial input is going to activate adjacent series of cells, and they're all going to feed in to one output. But what do these different circuits do? Well, for converging circuits, typically information is being collected. So we're collecting stimulus over a large area and feeding that back to the brain, for instance. And the best example I can think of that occurs within the eye. The diagram that you're seeing here shows rods within the eye. So in the eye, you have photoreceptors, which are specialized nerve cells. They can fire off an action potential. And we have rods that are very sensitive to low light. They're what you use at night. And then we have cones that can sense different wavelengths of light. So they're responsible for color vision and also high acuity or high resolution vision. What you see here is we have 3 rods that feed into one bipolar cell, and then that bipolar cell feeds into one ganglion cell that leads back to the brain. Now don't worry about the different cell types or their names here. It's not usually just 3 rods, it's usually hundreds of rods. So hundreds of rods can feed into one bipolar cell. Rods are very sensitive, and if just one of them collects a photon of light, that might be enough to trigger the bipolar cell and send a signal back to the brain. So you can see we're collecting very weak stimulus over a large area. Now, because of that, if you're using rods for vision, you get a very pixelated view of the world. Cones, on the other hand, have a 1-to-1 ratio when it comes to the cone and the bipolar cell, and you get higher resolution, you get smaller pixels in your vision. Diverging circuits typically are involved in outputs. So a nice example might be the cells within the brain stem, that control breathing. There's a small collection of cells there. One of them can send off a signal that will go to the lungs, and it will be spread out over several cells that can affect the breathing rate for that entire large organ. So we have a small output from the brain that can stimulate a lot of cells. With reverberating, this is a pathway that's involved in memory. So if you're doing anything, if you're saying a sentence, if you're adding things together, you have to remember what you've just done. If you start a sentence and you can't remember the last word you said, you can't finish that sentence. This is short-term memory. Here what we have is this reverberation. So this pathway is kept active. The cells that are in the end of the series activate the ones at the beginning, and this appears to play a very important role in maintaining short-term memory. Parallel after discharge, remember this is where we have these parallel series of cells that are sending a signal, and what that parallel system does is it makes sure that we maintain that pathway. If one of those pathways stops working for some reason, we have adjacent pathways. These systems appear to play a rather important role in cognition, thinking. A circuit that includes a sensory input and then a motor output is termed an arc. So we have our afferent neuron that's carrying information away from a sensory structure towards the central nervous system. And then we may or may not have integration occurring within the central nervous system. And then we have an efferent nerve that carries information away from the central nervous system to bring about an effect at an effector. Now, it might surprise you to learn that this sort of arc doesn't necessarily have to involve the brain. There are a lot of very simple, quick, automatic responses, things you don't have to think about, that only involve the spinal cord or the cranial nerves. That's something referred to as a reflex. So a reflex is a fast, predictable, automatic, unconscious response to a change in the environment that's going to help maintain homeostasis. We can have reflexes that involve the spinal nerves. We can have reflexes that involve cranial nerves. So blinking, for instance, if someone brings their finger close to your eye, you blink, you can't help yourself, or at least it's difficult to prevent yourself from blinking. That's a cranial reflex. We can also have the autonomic system involved, and we'll come back and spend a bit of time on the autonomic system. Reflexes can be somatic. That means that they involve skeletal muscle. or they can be visceral, which means they involve the internal organs. A reflex arc, the simplest pathway, has to include at least one synapse. So at the very least, we have to have one afferent neuron and one efferent neuron. So we have a sensory input and we have a motor output. Now we may have one or more interneurons in between. And these pathways involve the spinal nerves and also the spinal cord itself. And these kind of reflexes allow for very rapid responses without requiring the brain, so it's not something you need to think about. And quite often these responses are to avoid damage to the body. So for instance, if you touch something hot, you immediately pull your hand away. You don't have to think about that. Here we have an example of a very simple reflex arc. There's only 3 cells involved here. We have our sensory receptor that runs from the skin, in this case, back to the spinal cord. Then we have an association neuron, also known as an interneuron. And then we have our motor neuron that heads back out to muscles. Let's walk through what happens in this simple arc. So let's say that you're walking around barefoot and you step on a piece of glass or you step on a tack, something like that. You damage your skin, and that's going to set off action potentials within the free nerve endings of this sensory neuron. It's quite fascinating how this works. There's a few different pathways that can be involved, but the simplest way it can work is that we just damage that neuron. That pokes a hole in the plasma membrane of that neuron, and that allows sodium to rush in, and that depolarizes the cell. But in any case, we get an action potential that rushes back towards the spinal cord. Remember that the cell bodies of these sensory neurons are located within the dorsal root ganglion of the spinal cord. Now that signal gets to the spinal cord, and then within the gray matter of the spinal cord, we have these association or interneurons. It's going to synapse with that neuron, cause it to fire off, and then that is going to cause a motor neuron to fire. That motor neuron is going to carry an action potential back out through the ventral root, back to the spinal nerve, and out to muscles that will cause you to lift your foot and move your foot away from that source of damage. Now realize that the pathway we're looking at doesn't involve the brain. All of this thinking, quote unquote, is being done by the spinal cord. It's a preset pathway. Now that association neuron will actually synapse with other cells. It's not shown here, but it will synapse with other cells that will carry information up the spinal cord to your brain, which will make you go, ouch. Reflex arcs come in a few different forms, so I'm going to throw a bit more terminology at you here. First of all, we can have what's called a monosynaptic reflex arc. Mono means one, so there's only one synapse. So in this case, we only have 2 cells. We have our sensory cell, and it will directly cause a motor neuron to fire. We can have polysynaptic reflex arcs. In this case, again, we have our sensory input, we have our motor output, but we have 1 or more interneurons. So the arc that we just looked at, there was 1 interneuron. This is the simplest polysynaptic reflex arc we can have. We can have ipsilateral reflexes. In this case, sensory information is coming into the spinal cord, and then the motor output is leaving the spinal cord, and this is all happening on the same side of the body. We can also have contralateral reflexes, and you've probably guessed what this is going to be. This is where we have sensory input and motor input occurring on different sides of the body. The somatic reflex that you're likely most familiar with is the patellar reflex. Remember that the patella is the kneecap, and it's suspended within the patellar tendon. If you take a reflex hammer, and maybe you've had a chance to do this in the lab, if you take that hammer and you smack someone on their patellar tendon, they will kick you, whether they want to or not. This is a stretch reflex. What's happening is when you hit that tendon, it's stretching muscles that make up the quadriceps group, the muscles that are used to extend the leg. Those muscles stretch very quickly. Your body, in response, extends the leg to limit that stretch, because a sudden stretch in the muscle might damage the muscle. So it's to prevent injury from overstretching. This is an example of a monosynaptic ipsilateral reflex arc. So it's monosynaptic because we just have a sensory input and a motor output. There's no interneuron involved in the contraction of the muscles that make up the quadriceps group. It's ipsilateral because it only happens on one side of the body. All of the cells that are involved are on the same side of the body. Let's take a look at this arc in action. So we take our hammer, we hit the patellar tendon, and what that's going to do is it's going to stretch the muscles that attach to that tendon. Within the muscles, we have these special sensory structures known as muscle spindles. They sense stretch. So when that is stretched, the sensory neurons that are connected to it will fire off action potentials. And you can see the sensory neuron here in blue. So the action potential makes its way back to the spinal cord. Now, several things happen within the spinal cord. We're going to look at the bottommost connection first. So notice that that sensory neuron connects directly to a motor neuron. That's shown in red, the red solid line. That's going to carry a signal back to those same muscles and cause them to contract. That's going to extend the leg, and that's going to limit the stretch. Now there's something else going on, and this second part is not a monosynaptic reflex. First of all, we have a signal going up to the brain. That's going to tell you that you just kicked your leg. But we also have a signal that goes to an interneuron within the gray matter of the spinal cord. This is an inhibitory interneuron. What it's going to do is it's going to fire off a signal that also is carried back towards the muscle, but this time to the antagonistic muscles. These are the muscles on the other side of the femur. These are muscles that you would use to bend your knee. This motor neuron is going to release inhibitory neurotransmitters, and it's going to make it impossible for that antagonistic muscle to contract during this contraction of the quadriceps muscles. So that means that you can kick your knee out without any resistance from these antagonistic muscles. Let's go back to the example I mentioned before of touching a hot surface. Imagine that you accidentally touch a hot stove with your finger. Now what's going to happen is you're going to instantly withdraw your hand and withdraw your arm. You don't have to think about it. It doesn't involve the brain. Thinking about it would take too long. And of course, the goal here is to prevent further damage. This is something known as a flexor or withdrawal reflex. Flexor because you're flexing the arm. You are contracting the biceps and other muscles that allow you to flex the arm and pull it out of the way. That's a fairly simple pathway. It all occurs on the same side of the body, so the sensation and the movement is occurring on the same side of the body, so it is ipsilateral. But we can have what are called crossed extensor reflexes. This time, let's imagine that, once again, you step on something sharp. So you step on a piece of glass. Now, we've already talked about how that's going to cause you to pull that limb away. So you're going to pull your foot back. However, that's going to put you off balance. So at the same time, your other leg has to move forward. It has to extend to maintain the balance. This is a contralateral arc. Let's take a look at what happens when you stub your toe. There's a fascinating series of events that occurs that doesn't involve your brain. First of all, we have a flexor withdrawal reflex. That's the reflex that's going to cause you to flex your knee and pull your foot away from the source of pain. On top of that, we have a contralateral extensor reflex, and that's going to cause you to extend your other leg to maintain your balance. We'll come back to that one in a second. Let's start with the flexor reflex. So you step on something sharp or you stub your toe. that's going to cause a sensory neuron to send a signal back to the spinal cord. Now note that this reflex doesn't just involve a single spinal nerve, it involves three. So we're seeing three sections through the spinal cord. This is an area of the spinal cord that would be covered by three vertebrae. Anyway, so our sensory signal is going to go back to that innermost or middlemost section of the spinal cord, and this sensory neuron is going to synapse with an interneuron. That interneuron is going to synapse with a motor neuron. That's going to send a signal back to our flexor muscles that can withdraw the foot. But this one signal is not enough. We have to stimulate the muscle over its entire length. So note that we have other interneurons. We have an interneuron that can send signals, action potentials, both up and down the spinal cord. So I'll outline that for you. We have this interneuron here that can send a signal up to the next section of the spinal cord, and we have signal being sent down by that same interneuron to a lower section of the spinal cord. Same thing is going to happen in those spinal cord segments. We have an interneuron that will be activated, and that will activate a motor neuron, and that will send additional signals back to the flexor muscles so that we can get a powerful flex of the muscle that will pull the foot away from the source of pain. This is all ipsilateral though. Note that it's all occurring on the same side of the body and the same side of the spinal cord. And because that wasn't complicated enough, let's throw in our crossed extensor reflex as well. So on the left side of the diagram, we have the flexor reflex arc that we just talked about, but note that we also have interneuron connections that go across the gray matter within the center of the spinal cord. They go across the gray commissure, which hopefully you've had a chance to see on slides in the lab. It's a thin little connection of gray matter. These connections are going to carry signals to the opposite side of the spinal cord, and these interneurons are also going to synapse with interneurons that carry information up and down through the spinal cord. The end result is that we have three motor neurons that have been activated on the other side of the spinal cord, and they're going to carry signals to extensors of the opposite leg. So that's going to cause the opposite leg to extend. The foot is going to be moved forward, and that's going to ensure that you're not thrown off balance. Of course, if you just suddenly withdrew your leg and you didn't compensate for that, you would fall over. It really is quite remarkable what the spinal cord can do all on its own without any input from the brain. Remember that the whole point is that things be done quickly. If you had to take the time to think about an appropriate response, well, it might be too late. The damage would be done already. It's not shown in those diagrams, but there are other interneurons that are involved that will send signals up to the brain to inform you about the stupid thing you just did. And we'll talk about that when we talk about the brain in our next topic.